finished the conversation about the blood, and I want to remind you, we had this little talk, little chat about clotting. So we mentioned a couple of clotting diseases: thrombocytopenia, generally lack of platelets or low numbers of platelets; hemophilia, specifically the abnormality of clotting factors, genetic absence of those; and disseminated intravascular coagulation. Now, you can easily see that these are the pathologies associated with an ability of blood to clot. Sometimes ability of blood to clot becomes actually an issue, and the conditions such as deep vein thrombosis, atrial fibrillation, higher propensity of blood to clotting can cause a bunch of troubles, from stroke to myocardial infarction. So there are drugs that inhibit clotting, and aspirin being the first one. Okay, so aspirin is the inhibitor of thromboxane A, and we mentioned before that thromboxane A is one of the key We mentioned before that thromboxane A is one of the key mediators in platelet activation. So if we inhibit thromboxane A, we essentially inhibit platelet activation, we inhibit formation of the platelet plug. Now, aspirin used to be prescribed to patients with a high risk of thrombosis. At some point, actually the recommendation was to just take aspirin if you are above a certain age. Recently, American Heart Association revoked this recommendation, saying that the risk-benefit ratio is not justified for healthy people. The risk of internal bleeding is much higher if they don't have the underlying thrombotic conditions. Does that make sense? Now, heparin, just anti-thrombotic drug. Blood transfusions used during the surgery to prevent unnecessary clotting can be added to the blood that's drawn from the patient to prevent clotting, widely used. Now, warfarin and Coumadin. Warfarin actually is a component of a typical rat poison. If you watched the series House MD, in one of the episodes, House was trying to kill a rat using warfarin, antithrombotic drug. It is actually used in patients with atrial fibrillation, which gives you an idea that the dose makes the poison. It can be a poison or it can be a drug. The mechanism is that interferes with the effects of vitamin K on coagulation process. The newest drugs that prevent thrombosis belong to direct thrombin inhibitors. So this image shows you these drugs. So they directly inhibit thrombin, preventing the conversion of fibrinogen into fibrin. So this image actually shows you a bunch of antithrombotic drugs, but the only ones that I want you to kind of recognize this are gatroban, hiridin, and dabigatran. So these three drugs directly inhibit the function, the enzymatic function of thrombin, preventing the conversion of fibrinogen into fibrin. Now, the drug called hiridin was isolated from a small blood-sucking animal. Leech, yes. And actually, leeches are a legitimate therapeutic approach. Just, let's not go back to 19th or 18th century thinking that leech therapy can treat anything. Leeches, sterile leeches, are used to remove clots before reattaching the severed limbs. Leeches are basically applied to the site of injury, and they will essentially remove the clots from all those vessels that were damaged. Does that make sense? They increase the blood flow in the damaged limbs. Now that concludes our conversation about blood and brings us to the talk about the immune system. And this year I decided to kind of cut down a little bit on the immunity in AP2 because it's kind of a funny situation. There are two classes, biology classes that we teach here that teach immunity. It's AP2 and micro. And in my opinion, it is kind of more, I teach it more in depth in micro. So here we're going to focus more on a general patient outcomes and basic mechanisms. In micro I do a bit more in depth. Okay, so I kind of reshaped it a little bit to save some time and space. So immunity involves several types of barriers. Terminologically, there is a mess in what we call different branches of immunity. There are like intrinsic immunity, innate immunity, adaptive immunity. I tend to refer to intrinsic barriers as chemical and mechanical barriers that protect various body sites. And so if you think about, okay, let's talk about mechanical first. The main mechanical barrier that we have is the skin. That's the entire organ, right? Bless you. It's kind of obvious. Do we have anything more subtle? The answer is yes. Mucus. Any mucous membrane, if you think about it, mucous membranes are always exposed to the outside. Your respiratory mucosa, your digestive mucosa, your urethral mucosa, they all have the potential to interact with the environment. Does that make sense? So all these mucosal surfaces, from mouth to genitals, they're all going to have mucus, which functions as the pathogen entrapment. Not only this, in many locations, that mucus will be moved, especially in the respiratory tract, by beating of cilia. So mucus in your trachea, for instance, serves as some kind of a glue for dust, pollen, any kind of debris, any kind of microorganisms. And then these entrapped microorganisms are propelled upwards towards the split between the larynx and the esophagus, the trachea and the esophagus. They propelled there by the beating action of cilia. Now what about the chemical barriers? Probably the most notable chemical is lysozyme, which can be found in virtually any body secretion, from tears and saliva to just mucosal secretions. Lysozyme is a broad-spectrum antibacterial protein that essentially kills bacteria. Does that make sense? And that what controls bacteria that are normally present in your eyes or in your nose and stuff like that. Dermicidins, as you can guess from the name, are unique to skin, derm. They're produced in the sweat and protect your skin from bacterial invasions. A large portion of chemical barriers is the altered pH. For instance, your skin is slightly acidic. Stomach is very acidic. Vagina is quite acidic. So all this acidity creates sort of an unfavorable environment for microorganisms. Moreover, if you think about the urinary tract, which we didn't talk about yet, urine is slightly acidic, having pH around 5. And that lower pH actually makes urethra quite an unfavorable place for microorganisms to grow. Does that make sense? There are reports that some fatty acids on the skin in the sebum actually have some antibacterial properties. But like very brief but important mention just to give an idea. A lot of locations in the human body have their own microorganisms, resident microorganisms. Skin, digestive system, vaginal mucosa, parts of the respiratory tract. All this microbiome effectively inhibits the invasion by increasing the competition. That makes sense? And that, in terms of importance, it's probably comparable with any other branch of immunity. Does that make sense? So know your chemical and mechanical barriers. Now we're going to go back to the cells of an innate immune system and review them fairly briefly. So first we need to define what is innate immunity. Innate immunity is already present. It is ready to protect from the get-go. It doesn't require an exposure to the pathogen to get activated. Unfortunately, that makes the system fairly nonspecific, and that lack of proper specificity limits the immune system, the innate immunity effectiveness. Okay? The advantage, of course, is that innate immune response is very rapid and can start in the matter of an hour. The cells that mediate innate immune responses can be divided into two large groups, as we discussed before. Those are all white blood cells, granulocytes and agranulocytes. And in this table, the granulocytes are highlighted by beige and agranulocytes by pink. There are four main types of granulocytes that participate in the innate immune responses. First is neutrophils. Sometimes in the clinical reports and medical literature, you can see the acronym PMN. PMN stands for polymorphonuclear, meaning that the nucleus has this weird multi-lobed shape. Neutrophils are called neutrophils because they can be stained microscopically by both acidic and alkaline dyes. And they belong to granulocytes since they contain cytoplasmic granules. You can see granular appearance of the neutrophil cytoplasm on the image. Neutrophils are the most abundant white blood cells, and their main responsibility is to fight off bacterial infections. They can do so through phagocytosis. Okay, and we're going to talk about mechanism of phagocytosis. They can also release the chemicals of respiratory burst. Now, these chemicals are strongly oxidative, such as hydrogen peroxide or different chlorine compounds. This chemical, essentially, they imagine that these neutrophils spray bacteria with the bleach and hydrogen peroxide. That's essentially what happens. Another fascinating mechanism by which neutrophils can limit bacterial infections is called NETs. NET stands for neutrophil extracellular trap. Neutrophils are essentially the cellular kamikazes. They die when they protect. When neutrophil dies, it releases its DNA. Neutrophil DNA wraps around bacterial cells and essentially tethers them to one spot, preventing the spread of the infection and allowing the chemicals of respiratory burst to work more effectively. That makes sense. Now, eosinophils can be stained by acidic dyes, and the main targets are eukaryotic pathogens like fungi, protozoa, or helminths. So this clear distribution of targets allows you to get some hints about the diagnosis of infectious disease based on the prevalence of the cells in the blood. If you take a blood draw and you do the white blood cell count and you see quite pronounced neutrophilia, meaning a lot of neutrophils, you can conclude that it's most likely bacterial infection. If you see eosinophilia and you suspect infectious process, it's either protozoa or helminths. Fungal infections are just very rare. Okay, you can have this distinction. You see what I'm saying? Now, eukaryotic pathogens are large. Some of them are even multicellular. Take helminths, the worms, they're big. So eosinophils do not phagocytose. Instead, they produce toxins, either chemicals of respiratory burst or specific factors that are toxic for fungi or protozoa or helminths. They may not even kill microorganisms, but especially for helminths it's true. These toxins can actually force helminths to leave, like bug them off, okay? Does that make sense? Eosinophils are also associated with allergic reactions. Studies, for instance, found that in patients with asthma, you can find higher numbers of eosinophils in the tracheal tissue compared to patients without asthma. So it's likely that eosinophils do mediate some aspects of allergic response. Make sense? Basophils, called basophils because stained by alkaline dyes. You can see basophil here. They're very granular. See multiple dark granules. And, well, clinically, you will see basophilia in allergic reactions. However, of course, allergy is not a function, it's abnormality. Does that make sense? Basophils are basically cells responsible for inflammation. They contain enormous amount of histamine, and they will release histamine in the sites of injury, increasing vascular permeability and increasing the local leukocyte recruitment. Does that make sense so far? Now, all these three types of granulocytes can be found in the circulation. Now, you can take a blood draw and very easily see a bunch of neutrophils. Eosinophils and basophils are much less frequent. Now, the fourth type, a mast cell, is basically a basophil that went into the tissues and became resident there. So mast cells reside in tissues, and these ones participate in allergic reactions. As you will see, when you have an allergic reaction, say you have allergic rhinitis. Anyone is sensitive to pollen in the spring? I guess so, some folks, yeah. So You go out there, you get exposed to the pollen, and mast cells in your respiratory mucosa start to degranulate. They release histamine. What you do, you take antihistamine. You take Zyrtec or whatever. I forgot the second one. See something. Anyway. So you take the drug. That makes sense? Which inhibits the histamine release. And ideally, you take it before you go outside. So mast cells do not degranulate. Are we clear about granulocytes? Once we wrap up the cells, I will tell you what you need to focus on. Good? So what about agranulocytes? First, we're going to talk about monocytes and macrophages. You can see a monocyte in the image, in the upper right corner. And as you can guess from the image, this monocyte is surrounded by red blood cells. Monocytes can be found in the circulation. They are largest white blood cells. And they are dysfunctional. Meaning, monocyte itself doesn't do much. But when monocyte is exposed to inflammatory response, it becomes a macrophage. It doesn't, bless you, doesn't change the shape that much, doesn't change the appearance that much. I'm sorry. But the difference is macrophage, which looks just like monocyte, starts to migrate into tissue. Because this is where the injury happens. This is where the problem is. You see what I'm trying to say? So monocytes are like Bruce Banners running around in circulation, and then when they're exposed to some kind of inflammatory stimulus, they become incredible hulks and move into the circulation. We mentioned it before. So macrophages, a main function, macrophage, the large eater. They phagocytose cells. They phagocytose dead cells. They phagocytose bacteria, viruses, I don't know, anything virtually. Now, there's one more type here. One more cell type in this table that can also phagocytose. Can you see that? It's the very first line, the neutrophil. So neutrophils and macrophages are two phagocytic cells. You clear? The main difference between them, neutrophils, when they phagocytose, they die. That's it. Macrophages can just keep going. They will munch on whatever they're supposed to and just keep moving, and they will be eating more and more and more and more cells, processing them and destroying them. Does that make sense? The last type of cells that I want to mention here is the natural killer cell, and it's quite a fascinating cell. So that's natural killer cell. It's pretty small. And essentially, natural killer or NK cells belong to a distinct population of white blood cells called lymphocytes. A while ago, we were talking about two lineages of white blood cells. Do you remember that? We mentioned that there's a myeloid lineage, like blood cells, and then there is lymphoid lineage, cells that will go into the lymphatic system. So NK cell belongs to lymphoid lineage. We clear so far? Now from the name natural killer, you can gather the idea that it does kill something, and it turns out it's sort of specific against virus-infected cells or cells with cancer. Now, cells that are infected with viruses are cells that are malignant, cancerous. Okay, let me start over this. Cells, our cells, have special markers on the surface. Are you with me? Like when you play, I don't know, a game of, when you play football, your team wears certain colors, and opposing team wears different colors, right? So your cells wear sort of standard colors. And if your cell gets infected or becomes malignant, this quote-unquote colors, the surface markers, they change. NK cells can recognize that change and, basically saying, oh, that's bad. That's not ours anymore. We're going to kill it. Now, NK cell, you can say, well, hold on, that's quite specific. Well, it's still innate immune response, because NK cell does not differentiate between cell that is cancerous or cell that is infected with measles or cell that is infected with rickettsia. For NK cell, these cells are bad for it, all of them. That makes sense? So that lack of specificity is what makes NK cell a part of innate immunity. Now, how does NK cell kills those cancers? Two main mechanisms. Well, one mechanism. This mechanism is apoptosis. So NK cell is kind of a magic, has a magic wand. It touches cancerous cell, and cancerous cell commits suicide, basically. Controlled, very well controlled suicide. You with me? Now there are two ways for NK cell to do that. One way is called Fas ligand Fas receptor system. So you have NK cell, you have target, okay? And crap, I never remember. I'm not going to ask you this, but it's just so embarrassing. I don't remember which one expresses ligand, which one expresses the receptor. I believe that whatever. Say Fas receptor is on the target, Fas ligand on the infected cell, they interact, that initiates the signaling in the target cell dies. Does that make sense, the idea? Just ligand receptor interaction. Second mechanism is so-called perforin granzyme mechanism. So I'm going to introduce it briefly because we're going to talk about it again. Let's say red is the infected cell and blue is the NK cell. Okay? So NK cell will release proteins that are called perforins. Based on the name, what do you think they do? Perforins. Perforate. That's like why they gave them this name. They perforate. They perforate the cell membrane, forming pores. And then enzymes called granzyme, they enter the quote-unquote bad cell and initiate apoptosis. Does that make sense? So both mechanisms, the ligand receptor mechanism that I pictured here, or Fas ligand Fas receptor mechanism here, they both end up with apoptosis, but the way they achieve that is different. Any questions?
What are the green arrows that
Just granzymes.
Okay.
I'm going to circle the granzymes and perforin words. So granzymes in the right bottom corner. So greens are granzymes, and the blue are perforins. What you need to know: where you can find the cells, whether they are granulocytes or agranulocytes, their main targets, main mechanisms. So basically I can ask you, which of the following cells can phagocytose? We have two. That's easy to remember. Which of the following cells protects from bacterial infections? Neutrophils. Does that make sense? That's kind of general idea. Any questions? Yes? So, which group these cells belong to? where you can find them, the main targets or functions, and mechanisms of protection. So say that's the last column, just general idea what they can and cannot do. So you have only 2 phagocytic cells. Okay. We're clear? Good. Perfect. I also have a little request. Can we do a break that early? Like not at like 12:40, but maybe 12:25. Is that okay with you? Just need to catch a person in a very short window. So okay, leukocyte recruitment, we discussed it before when we talked about white blood cells in the blood discussion. Now we're going to chat about phagocytosis. So it's rather non-specific. So phagocytes, macrophages. Let's talk macrophages for a second. Remember we were talking about congestive heart failure. And I mentioned that if left side fails, the patient will have pulmonary edema, right? And red blood cells will actually end up in the lungs, and pulmonary alveolar macrophages will phagocytose them. There was a conversation about it. And that tells you they will phagocytose anything. If it's not in the right place, they will munch on it. If it's dead, they're going to munch on it. They're not really specific. Does that make sense? However, they can tell apart. They can say, they can see normal live cells. See what I'm saying? So they will say, okay, this cell is live, this cell is not infected, it's not bacteria, it's not fungus, it's not protozoan. Pass by. Okay? Cells have... Give me an example of it's not supposed to be there. Good.
Like what you were just saying, like the accumulation of blood in the lungs. Like it's not affected.
Oh, no, it's they're not alive anymore, those red blood cells.
Oh.
They dead. Oh, it's going to just get rid of the... So those red blood cells are not functional. When they end up in the lungs, it's not only not supposed to be there, but they're not functional. Does that make sense?
Yeah.
If we hypothetically would be able to grow a chunk of liver in the lungs, I would bet macrophages would be just fine. Make sense? So they still can tell apart what's supposed to be phagocytosed from what's not. And when they find, say, a pathogen, they adhere, form pseudopodia, false legs, and engulf the pathogen or debris into a vesicle that's called phagosome. Phagosome fuses with intracellular vesicle called lysosome. Lysosome contains proteolytic enzymes, various oxidants called reactive oxygen species. So the idea basically is that chemicals from the lysosome will chemically destroy the content of the phagosome. Such fused vesicle is called phagolysosome. Okay. Once everything is destroyed, you have debris, which will be, the small... They either going to be used inside the cell, say, if it's a bunch of amino acids, we can put them to good use, right? Or, if they are not usable, they will be released by exocytosis. Now, again, I want to highlight, we have 2 phagocytic cells, neutrophils and macrophages. Neutrophils will die as the result of phagocytosis, while macrophages are going to survive. Any questions? Now, another component of innate immune response is called complement. It's an interesting system of blood proteins that, once activated, provide a certain non-specific immune responses. So first of all, what can activate complement? There are three activation pathways. In classic pathway, complement is activated essentially by antibodies bound to an antigen. Some of you may already know what antibodies are. If not, we will learn about it today. So antibodies bind to the antigen, and then antibody bound to an antigen. That's important. Antibody has to be bound to an antigen. Antibody has to be bound to a bacteria, for instance. And only after that they will activate classical pathway. Another pathway called lectin pathway is when a sugar, mannose sugar, on the surface of the pathogen, mannose on the surface of the pathogen, binds to a lectin molecule. So this mannose-lectin interaction activates the lectin pathway. And finally, the alternative pathway can be activated by some molecules on the surface of the pathogen. Okay, so we've got classic, we've got lectin, and we've got alternative, and you need to know what activates each pathway to the extent we discussed. Now, what's the result of that activation? Complement proteins show the same mode of action, similar mode of action as coagulation proteins do. Meaning, one protein activates, so number 1 activates number 2, number 2 activates number 3 activates number 4. Does that make sense? This is why we call many of these responses a complement cascade. The only thing that you need to know about the complement cascade is that the result of classical pathway activation or lectin pathway activation or alternative pathway activation, the eventual result is the enzyme called C3 convertase. So these three pathways, they essentially converge at this C3 convertase. We clear? And from C3 convertase, you're going to have your protective outcomes. So one outcome is the general stimulation of inflammatory response and recruitment of red blood cells. Rather non-specific. Does that make sense to you? Second outcome, shown here, is called opsonization. Now let's talk about opsonization a little bit more. So imagine that I am a bacterial cell. Okay? And I blend in quite nicely. Okay? What complement does, it's like a tag that says danger or bacteria or something like that. Basically it says danger. Does that make sense? So complement proteins, they attach to the surface of the bacteria, making it more attractive to macrophages. Make sense? So bacteria used to be able to, quote-unquote, hide, but with complement proteins on it, it's extremely noticeable and very attractive for phagocytosis, and macrophages and neutrophils will just remove it. Does that make sense? That's opsonization. Now, make a note about opsonization. It will be very useful later. Opsonization is not necessarily a process that only complement can participate in. Anytime, any molecule that sticks to a pathogen and makes it more attractive for phagocytosis, just to make a pathogen more attractive for phagocytosis, we call it opsonization. Does that make sense? When complement does it, well, complement-mediated opsonization. Is that good? Is that clear?
Can you repeat that?
So what I'm trying to say is that opsonization is basically making a pathogen more attractive to phagocytes. And one of the mechanisms to do that is to activate complement so that complement proteins will bind to the surface of a pathogen and stimulate phagocytosis. Better? Cool. The third mechanism, one of my favorites, is the formation of membrane attack complex. So C3 convertase leads to activation of 5 proteins, which, like in a Voltron cartoon, form a pore. You see the pore? And that pore will let water to go in or out, ions, whatever. Basically, it ruins the permeability of the membrane and kills the cell, leads to lysis. And you see, since it's a membrane, we call it membrane attack complex. So complement can actually lead to a lysis of the cell. Does that make sense? We're good? Everybody's keeping up? Now, the last mechanism of innate immune response that I wanted to discuss with you is interferons. Interferons are the proteins produced by the cell in response to infection. Okay? So look at this. That's virus. Can you see the virus? On top. Here. Virus. Virus gets into the cell. Okay? Now the question is, can cell recognize that it's infected? The answer is yeah, it can. Well, I mean, let's not make cells too anthropomorphic, but it kind of helps. So, cell recognizes that it is infected, and the infection activates the synthesis of interferon proteins that are released from the cell into the extracellular fluid. Interferons can exhibit autocrine activity, acting on the same cell. Basically, cell that produced interferons, you see what I'm trying to say? Cell that's produced interferons will also respond to interferons. And that's the cell, it produces interferons, they will act on the same cell. That's autocrine, right? Interferon may act on the adjacent cell, like in this image, okay? So they will all time, every time, interferon acts on the cell, it has to bind to its receptor. You see interferon here binds to the receptor. That's adjacent cells, that's paracrine action. Or interferons can enter the bloodstream, be delivered to some distant site, and act on the distant cells. That's a typical endocrine action. So we can technically consider interferons being some kind of a hormone, really. I mean, it's not produced by a specific gland. Make sense? So now, presumably you have no virus infections. You're not producing interferon. Does that make sense? When you get infected, that's when you start. We clear? Good? Understood? Cool. Now, what happens when interferon binds to its receptor? It triggers antiviral state. So-called interferon-stimulated genes are expressed, and the outcome of this, it may prevent a successful infection, it may stop virus replication, all kinds of, it may actually kill the cell in response to virus infection. But it's good, because cell is dead, virus is dead. Does that make sense? You understand what I'm saying? There are two types of interferon, type 1, type 2. Basically, alpha, beta, lambda, and omega. I could never keep up with all those Greek names. They are type 1. Type 2 is gamma interferon. No matter what everybody else will tell you, type 1 is better. Just know that. Type 1 is better. It's the best interferon ever. Why? Because I was working on it. This is why it's better. Generally speaking, type 1 is produced in response to viral infections, type 2 in response to bacterial. Now, if interferons can stimulate sort of cells being resistant to infections, can we use them as a drug? The beauty of English language is that you have two different verbs for modality of action, can and may. Okay, or should. Oh yeah, we can. We definitely can use interferons as a drug, and we do use them as a drug, but we use them very carefully. Modulating such well-balanced system as immune system is almost impossible. Interferons are used to treat certain types of cancers. You know, you can see that. When somebody has a cancer, nobody really cares about side effects that much. Also some really severe infections. We're talking about something like a disseminated yellow fever infection, or Ebola virus infection, or in some cases people with really bad flu, like hospital requiring flu, you know, viral pneumonia, systemic infection at the brink of death. Yeah, then interference, because they have terrible side effects. One of the most prominent side effects of interferon treatment is depression. Clinical depression. It's one of the things that doctors watch really closely in patients. Does that make sense? So you can, but you got to be careful. Which interferons conclude our conversation about innate immunity, and we embark on the travel through the adaptive. So what's the difference? First of all, adaptive immunity is very specific. This is what the vaccines are built upon. When you get vaccinated, your adaptive immune mechanisms, they produce that immunological memory that allows you to fight off the same infection later with much higher efficiency. Does that make sense? This is why we call it adaptive, because it adapts. It targets itself against a pathogen. Sometimes I compare adaptive immunity to a really smart MMA fighter. You know, when you fight somebody that you never met before, first time, you don't really know what to expect. Makes sense, right? You don't know the person's, like hypothetically, you don't know the person's strengths and weaknesses. But you learn very quickly that, I don't know, this person is really good at grappling, but not so good at, I don't know, kickboxing, stuff like that. And next fight, you adjust your fighting style according to the person you're fighting with. That's your adaptive immunity. If you get influenza a second time, cells that will respond to that second infection with influenza, they influenza specific. You see what I'm trying to say? So, because of this, we need a exposure to the pathogen. And it means that it's going to be slower. Adaptive immunity takes time to develop. Weeks, basically. However, due to its precise, honed in nature, systemic adaptive response is more effective. So which cells mediate adaptive immune response? There are two types of lymphocytes that are generated in the bone marrow, the B and T cells. Now, B cells stay in the bone marrow where they mature, while T cells migrate to thymus, where they undergo maturation with the participation of thymus hormones, such as thymolin, thymosin, and thymopoietin. Okay? Once matured, B and T cells seed lymphoid organs. This is a strict term, seeding lymphoid organs. They migrate from thymus, if it's T cells, or bone marrow, if it's B cells, into the lymph nodes and spleen and reside there until they are exposed to some kind of an antigen. Exposure to the antigen activates certain populations of B and T cells, and upon that activation, B and T cells differentiate and proliferate, forming effector cells and memory cells. Now, effector cells, what's their function based on the name? What do they cause? Some kind of effect. They do stuff, okay? It can be killers, helpers, antibody-producing plasma cells, but they do something. And memory cells, they essentially form immunological memory. They will be responsible for you, quote-unquote, remembering the infection. Got it? And I want to say that effector cells and memory cells, they are in the blood. So at this point, no, lymphocytes are in circulation. That's it. They left the lymphoid organs. You don't need them in the lymphoid organs. You need them everywhere. And the only fluid that can deliver them everywhere is blood. Does that make sense? Good? Questions? We're going to talk about B cells first, then T cells, and we're going to chat about each step of development. But before we do this, we're going to talk about antigen and take a break. We need to define what it is. Antigen. Opposite origin. Antigen is large. Now, that's really some key characteristics. It is large and complex molecule that is foreign and can trigger the adaptive immune responses. So what can be a complete antigen here? Generally speaking, a complete antigen is protein. It can also be DNA or complex, large carbohydrates. Good? Is that understood what I just said? So complex, large carbohydrate. Now, antigen has many unique antigenic determinants. Now, can you imagine a molecule? No, seriously, like protein, like globule. Can you imagine that? And it has a certain particular shape, right? You see what I'm trying to say? Certain shape. And out of that shape, there are only some fragments of that molecule that are actually recognized by immune system. Oh, to hell with that. That's not a perfect analogy, but that kind of gives you an idea. Here's the deal. When you meet someone first time in your life, and you had a phone conversation with the person, and you need to meet with this person, and you ask, what do you look like? You know, what am I looking for when I'm meeting you? Person will likely not describe him or herself saying, one head, two arms, two legs. I mean, that's correct, but that's very generic, right? So the description will be I don't know, Indian's baseball hat, or I don't know, orange jacket, or something like that. You know, specific details. Or, you know, I'm missing one hand. Well, that's kind of very definitive, right? Does that make sense? So when we say antigen is recognized by your immune system, it's not that the whole thing is recognized, the whole molecule. No. Certain parts of that molecule that are called antigenic determinants. You see that? Each of this triangle here, this round shape, this weird shape, those are antigenic determinants. Antigenic determinants are the parts of the molecule that trigger the immune response. So it can have like 3 antigenic determinants. And each antigenic determinant will trigger its own immune response. Does that make sense? Now, I almost jumped out of my pants showing you how important it is that antigen is large and complex. Why? There are molecules that are legitimately large. This buckets on your tables, the green ones, they're plastic. It's a polymer. Maybe polyethylene, maybe PTFE. Okay. Now, those molecules, polymer molecules, are they large? Oh yeah, they're gigantic. Are they complex? No. They're really, really simple. And that simplicity explains why a large molecule like polyethylene cannot be an antigen. Does that make sense? You see what I'm trying to say? Large molecule that is not complex, it's not complex enough to stimulate the immune response. What about the small molecules? Well, some of them are just simply too small. And some of them are kind of a borderline, so say thyroid hormone. It's small, right? So it doesn't generate the immune response even if you stick it into the mouse. It turns out, if you take the small molecule, it's called haptene, and you attach to some large carrier, the immune system starts to notice it and produce antibodies against it. I almost compare it to, like you walk around your peers and you're not, size-wise, you're not very different from your peers. But if you hop on an elephant and ride elephant to Lakeland, you're going to be noticed. Like, no matter what you're wearing, no matter who, you're going to be noticed just because of the elephant. Does that make sense? So when we attach haptene to a carrier, we'll put haptene on an elephant. So immune system starts to notice it. We're good? You understand what complete antigen is? Large, complex? Let's take a break and I will try to catch that person that